Hello students, in this module we are going to see different calculation methods generally used in the pharmaceutical analysis. In pharmaceutical analysis, why we need calculations? We have seen in the previous modules that the pharmaceutical analysis is carried out for various applications including qualitative and quantitative analysis. So when it comes to the quantitative analysis, the calculations plays a major role. If you look at the application of pharmaceutical analysis, we are doing different quantitative analysis, including the determination and the estimation of quantity of drug, excipient, impurity in the pharmaceutical dosage forms. And the pharmaceutical dosage form comes like tablets, liquid orals, injections, etc. So when it comes to the tablet, we are saying that 200 mg of paracetamol in that particular tablet. Like that, when it comes to the syrups or when it comes to the suspensions, we are saying that 100 mg per ml or 100 mg per ml or 1 gram in 10 ml. Like that, we used to tell the strength of the particular product. So when we say the drug content in a particular product in a particular format, there is a requirement of calculations we need to do when we do the assay of the particular product, or we are determining the quantity of drug present in the particular product. Like that, it is applicable to the excipient or into the impurity. When it comes to the impurity, we say the levels of the impurity in nanogram or microgram level, or we call it as ppb or ppm level, parts per billion or parts per million level. So how we calculate these levels? For that, we need to have the calculations. Also, when we do the analysis, we used to have standards. So when you do titration, we have different calculations of molarity, normality. We prepare 1 molar solutions or 1 normal solutions of sodium hydroxide or sulfuric acid. How will you prepare those solutions? For that preparation of solutions, we need calculations. And when we do the chromatography, you have the peak area or peak height of the particular chromatogram, and which is corresponding to the standard what you injected and the sample what you injected. When you compare the standard and sample, the standard will be in the per ml this much microgram or this much nanogram or this much mg, and that will be used for the calculation of the unknown sample. So like that, there are so many calculations we need to do during the pharmaceutical analysis. So it is essentially we need to understand the basics of these calculations to carry out certain pharmaceutical analysis applications. We will see few of the examples to understand those in the upcoming slides. Apart from the examples, what we have seen in the previous slide, like the calculations required for the assays or drug content estimations or impurity estimations, we also do calculations for some of the assessments or evaluations, like the physiochemical property evaluation of like density, water content, etc. Also, for pharmaceutical doses, especially the solid oral dosage forms, we do the dissolution testing, for which the release of the drug from the dosage form will be calculated through certain methodologies. And sometimes we apply the linearity curves in the calculation of the unknown samples. And generally, these impurity levels are determined using the calculations will be used to control the levels of impurities in the particular drug or drug products. What are the different kind of calculations? In summary, we can see in pharmaceutical analysis, for examples, there are some basic calculations we need to do for the preparation of standards, preparation of solutions, etc. We need to convert the units. For that, we need to understand the units of different units and units conversion. For that we are going to see. And we see the titration analysis. Titration calculations are required. Then we do the assay of active pharmaceutical ingredient. and assay of drug content in drug products. Then we do dissolution release. Then we also do drug concentration estimation or the chemical estimation using linearity standards. And also we do certain calculations for different method validations. Before we go into the calculation methods examples, this slide shows some of the units with the names and how it is being denoted. Like kg is kilogram, g is gram, mg is milligram, micrograms, nanograms, liters, milliliters, microliters. So these are the common units which are used in the pharmaceutical calculations. This particular slide shows different units used and the conversion of units. For example, kilogram to gram, gram to milligram, milligram to microgram or ppm, and microgram to nanogram or ppb, and liter to ml, milliliter, and milliliter to microliter. So these are the conversion and how much gram is for particular kg or particular microgram, how many micrograms for particular mg is given in this particular table for the understanding. Now let us understand the molarity, how to prepare 1 molar solution or 0.1 molar solution or 0.5 molar solution for a particular chemical. So molarity is nothing but the number of moles of a solute dissolved in 1 liter of solvent solution, which is known as molarity. The molecular weight of the compound in terms of weight is dissolved in 1 liter of particular solvent or water. So which is the solution strength of that particular solution is called 1 molar. For example, the molecular weight of sodium hydroxide is 40. Then you take 40 grams of sodium hydroxide dissolved in 1000 ml of water will give you 1 molar sodium hydroxide solution. So the molarity is equal to the number of moles of solute divided by volume of solution in liter. For example, 0.1 molar means it is 4 gram of sodium hydroxide in 1 liter of water. So this is straightforward, simple understanding you can have for the molarity. Now we are going to see what is normality. How will you prepare one normal or X normal solutions of chemicals? Normality is described as the number of gram or mole equivalents of solute present in 1 liter of solution. When we say equivalent, it is number of moles of reactive units in a compound. So what is reactive units in a compound? When we say sodium hydroxide, it is having the formula of NaOH in which the reactive unit is OH minus. So the OH minus is 1 unit. Hence the mole equivalent of sodium hydroxide is 1. So accordingly, when you prepare the 1 molar solution of sodium hydroxide, you have to use 40 grams of sodium hydroxide in 1 liter of solution. When it comes to the 1 normal of sodium hydroxide solution, also will have the same proportion of 40 grams in 1 liter of water. So because the mole equivalent is 1. But in case of sulfuric acid, which is having a reactive units of 2 hydrogen ions are present in H2SO4. Hence, the mole equivalent of sulfuric acid is 2. So how you are going to prepare a 1 molar solution of sulfuric acid and 1 normal of solution of sulfuric acid in as an example in next slide. In this slide, we are demonstrating a calculation for the preparation of 1 molar and 1 normal sulfuric acid solutions. Before preparing the solution, we need to understand the purity of the sulfuric acid as well as the specific gravity or density of the sulfuric acid. 1 mol of sulfuric acid, concentrated acid, will weigh around 1.8 gram. The density will be 1.8 gram and the molecular weight is 98 and the purity is 98%. So these all factors we have to consider for the preparation of the solution. So when you apply this specific gravity or density, it will give you the gram per ml conversion. So as we have seen in the molar solution preparation, the molecular weight equivalent grams, that is 98 is the molecular weight. So 98 gram of sulfuric acid we have to consider, but it is a liquid, so we have to convert it to ml and there is a purity. So this also need to be taken into consideration. So to convert into ml first, 98 grams, we take 98 divided by 1.8 will give you 54.44 ml of sulfuric acid. And when it convert to the 98% impurity, it will give you 55.5 ml. So when you take 55.5 ml of 98% concentrated sulfuric acid in 1000 ml of water will give you 1 molar solution of sulfuric acid. Since the sulfuric acid having 2 reactive units, so it is 2 mole equivalent. So when you prepare one normal solution, you have to consider exactly half of the 55.5, that is 22.75 ml in 1000 ml water will give you one normal solution of sulfuric acid. So this is how you have to prepare 1 molar and or molar solutions and normal solutions of different chemicals for the titrimetric analysis. Now in this slide, we are going to see how to estimate a normality of the accurate normality of a particular solution using a titrimetric method. We are going to see an example of estimating the normality of a hydrochloric acid solution. So we are prepared one normal hydrochloric acid solution here. And now we are going to estimate the normality, accurate normality of the prepared solution. For that, you are going to use a formula, that is weight of the titrate by using which compound you are going to titrate your hydrochloric acid solution. Then what is the required normality, which is 1, equivalent factor, and then the volume of titrant which is going to be assessed. So now you are going to take 1500 mg of anhydrous sodium bicarbonate as a titrant in 100 ml of water, and you add the indicator 0.1 ml and titrate against one normal hydrochloric acid solution what you prepared until you get a faint pink color solution permanently. So now you have the conversion factor which is obtained from this, that is 1 ml of 1 molar hydrochloric acid solution equivalent to 0.05299 gram, that is 52.99 mg. And the titration value, the volume of sodium carbonate solution used is 28.5 ml. And the weight you used 1500, approximately 1500 mg. By using this value, you calculate the normality which is coming to 0.9954. This is the accurate normality of your hydrochloric acid solution what you used in the titration. So this is how you are estimating the normality in the titration methods. In this particular slide, we are going to see some more examples of for the estimation of molarity and normality. In case of sulfuric acid, how will you do? So here, in case of molarity, you have the required molarity in the calculation, and in case of normality, it will be the required normality. So here Other factors like equivalent factor will be coming to the picture, where in case of normality it will be 2 and in case of molarity it will be 1. And then during the titration you will get the volume of titrant and weight of the titrate used in mg. So the titer value will be 20.4 ml, which is for the molarity calculation, and in case of normality it will be 40.4 ml. So based on that, the calculated normality will be 2.0347 m normals, and the molarity will be 1.0154 molar m. So this is how you can estimate the molarity and normality of sulfuric acid. Similarly, other solutions also can be estimated by using these calculation and titrimetric methods. As we have seen in the pharmaceutical analysis, you can do the assay of the active pharmaceutical ingredient. And you can do the assay of the active pharmaceutical ingredient by various methods, including the titration method. In the titration method, how will you calculate the assay? The assay is equal to the titer value into normality or molarity of the titrant into equivalent factor into 100 divided by sample weight into 100 minus loss of drying or water content. Either one you have to use into R, that is nothing but average weight of the tablet or capsule or volume of the liquid sample you applied, or the raw material means 100 mg of sample you weighed means whatever the raw material you are going to use, that will be the R value. So the loss on drying, LOD or water content will be applied for the raw material and oral dosage forms to calculate the on dried basis or unweighted basis assay. So this is what the purpose of LOD or water content you use in the percentage assay. That's why it is called as unweighted on dried basis or unweighted basis. You will put it in the certificate when you calculate this assay and prepare a certificate. So this is how the assay of active pharmaceutical ingredient or drug will be calculated using in the titrimetric method. When we do the assay of active pharmaceutical ingredient in a particular dosage form, or directly we are assaying active pharmaceutical ingredient as a raw material, we have seen the example of titrimetric analysis in the previous slide. And also you can do UV visible spectrophotometric analysis, you can do the assay, and also you can do chromatographic analysis by HPLC, GC, ion chromatography with the different detectors, which we have already seen in the previous modules. So when it comes to the UV spectrophotometry or chromatography, what will be the calculations will be applied? That will be see in the next slide. When it comes to the color spectrophotometric method or colorimetric method, the absorbance, molar absorptivity will be used for the calculation. So the molar absorptivity will be obtained using the absorbance of the 1 molar solution with 1 centimeter path length. So the molar absorptivity is equal to sample absorption divided by light path length into concentration of the solution. This molar absorptivity generally obtained from the pharmacopoeias, which will be used directly for the analysis. Specifically, when it comes to the UV visible spectrophotometric analysis, the absorbance measured in the instrument will be used for the calculation, or the molar absorptivity will be also used for the calculation, or the linearity calculations also will be used. So here we are seeing the example of how will you use the molar absorptivity, which you have seen in the previous slide, for the calculation. Here the percentage assay is equal to sample absorbance into sample dilution into 100 into R, that is the weight of the tablet, divided by molar absorbed into sample weight. R is the equal to 100 for raw material assay calculation. In case of chromatographic methods like liquid chromatography, gas chromatography, or ion chromatography, you will have the chromatogram which is having the peak area. So we have to see the peak area, sample area, and the standard area for the calculation. So the content, drug content or the assay is equal to sample area into sample dilution into standard weight into purity or divided by standard area into sample weight into standard dilutions into 100. So 100 is used generally for the percentage calculations, and the rest of the values you can obtain from the chromatogram as well as the preparation. When you do the standard preparation and sample preparation, these dilutions will come into the picture, and the sample weight of how much you weighed for the standard preparation and for the sample preparation, that will be applied here. So this is how you do the assay of drug content using the chromatographic method. So you are going to do dissolution when it is comes to a solid dosage form like tablet, capsule, and during dissolution you are going to see the drug content in the release media. So in that case, a similar kind of formula will be used for the calculation. If it is a chromatography method, then you will go for the peak area, standard area and sample area, or in case of the spectrophotometric method, that is colorimetry or UV spectrophotometry or any other spectrophotometry, the absorbance will be used here. Either of one will be used for the calculation, and you will have the similar kind of formula to calculate the dissolution or the release of the drug at particular time point in the release media. So this will be one of the applications in the evaluation of the pharmaceutical dosage form when you manufacture or when you develop it. Now it comes to the linearity standards. How will you use linearity standards for the calculation of drug content or the estimation of drug content by using particular method? This linearity can be applied in spectrophotometric method as well as the chromatographic methods. And these samples of linearity methods, we have already seen examples of this linear already seen in the previous modules also. So how will you construct a linearity? So you will have a concentration and the absorbance or the peak area here, which will use to construct the linearity curve first. So this is linearity curve is prepared based on the standards generally, not the samples. So standards you will use to construct the linearity at different concentration. You will get the absorbance, and how you will construct the linearity curve. How the linearity curve will look like, we are going to see in the next slide. So when you plot the absorbance here on the y-axis and the concentration in the x-axis, each concentration will give the absorbance like this, and you are putting a trend line or the linearity curve you plot here, which will give you the equation. By using the equation, you can calculate the drug content, unknown drug content from the unknown sample. So for example, you are getting the absorbance of the unknown sample in the range of 0.5. So you plot this 0.5 here against the concentration you will get here. So 0.5, if you put the 0.5 value in the y, will give the x amount of drug present in the particular sample. So this value you will get from the instrument, and this value you will calculate from the formula y is equal to mx plus c from the linearity curve. In this slide, we are showing one of the typical example of how will you calculate the concentration of drug in a sample using the linearity curve. So the formula which is y is equal to mx plus c. So in this particular example, the absorbance is 0.423. That is the y value is given, and the concentration of the drug in the sample x is equal to y minus intercept divided by slope, which will give you the value of particular concentration, that is 242.56 microgram per ml. So this is what I explained in the previous slide as well. Apart from the calculations what we have seen, the basic calculations, we used to do a lot of other calculations during the method verifications and method validations, like the standard deviations, relative standard deviations are being calculated by using different values. What we get, the percent CV is being calculated, mean of the different values obtained also calculated, the recovery will be calculated. So these are the different parameters: precision, accuracy, recovery, mean. All these different summary statistics will be calculated when we do the method verification and method evaluation, and when we have a lot of data. So these are all the other calculations what we employ during the method verification and method validation in pharmaceutical analysis. So students, in this module you have seen the basis method of calculation, including the molarity, normality calculations, and also the calculations, assay calculations, especially for the spectrophotometric methods, as well as the chromatographic methods, and the linearity method adopted for the linearity calculations adopted for the chromatography as well as spectrophotometric methods. Hope this module is useful for the basic understanding of the pharmaceutical analysis, calculations involved in the pharmaceutical analysis. Thank you. Wish you good luck.